We have the next speaker, and Dr. Purohit, Dr. Jogeshwar Sachidananda Purohit. He's our colleague in assistant professor. He's an assistant professor at the Cluster Innovation Center, and he did his PhD from the Department of Zoology in 2009, and he has been working in various institutes like HIT Institute of Biotechnology, and then he was in the Department of Zoology at CMH College, Thane, Mumbai, and for the last 7-8 years he has been with the Cluster Innovation Center, and he has an interest in chromatin modification and remodeling and aging. So Dr. Purohit, we will be very happy to hear you, and we are running a little bit short of time,
Sure.
so you can help us.
Thank you, sir. Respected chairperson and the august audience. I feel happy and a little bit stressed today because I could see my PhD guide, who is also sitting in the audience, who actually introduced me to this exciting field of chromatin biology as a PhD student, which I am still continuing after getting a position in various institutes. So basically, the work which I am going to present today is directly not related to diseases, because I will be talking about chromatin and aging. Aging is directly not related, it is not a chronic disease. However, many of the chronic diseases and other diseases are associated with aging, but I will be talking about the relationship between chromatin and aging basically. So to introduce you with the chromatin, so the eukaryotic DNA actually associates with histone and non-histone proteins to give rise to a dynamic polymer which is known as chromatin. However, this polymerization or this polymer is not static or random, but while it is polymerizing, it compacts into different folds. Some of these domains are actually loosely bound or they are open, and some of these folds or some of these domains are tightly bound or they are more compact, and by virtue of that they give rise to heterochromatin and euchromatin, and they have differential transcriptional status. The euchromatins are transcriptionally more open than the heterochromatin. The differential folding is due to diverse kinds of post-translational modification which happens in histones. So all these 4 histones like H3, H4, H2A and H2B are subjected to diverse kind of post-translational modification like acetylation, methylation, phosphorylation and many more. So looking at the N-terminal tail of these 4 histones, one can understand that out of many of these modifications, few residues I want to highlight here. The lysine 4 of histone H3 and lysine 9 of histone H3 are of more important because they can be methylated, and their specific methylation at either 4th or 9th residue gives rise to different consequences which are downstream. Like methylation at K4 or lysine 4 ultimately gives rise to an active chromatin or an euchromatin domain by recruiting subsequent histone acetyl transphorases and other modification machineries. Similarly, methylation at lysine 9 subsequently recruits many of the other chromatin modifying enzymes, and subsequently it also methylates DNA. It is responsible for methylating DNA by recruiting DNMT or DNA specific methyltransferase, and it ultimately gives rise to an heterochromatic domain. These epigenetic marks which we call about, or the modifications in histones, they differ from a young cell to an aged cell. So while moving towards aging, or when a cell ages, there is change in the epigenetic marks altogether, starting from there is a decrease in the core histone expression to change also in the epigenetic signature or histone modifications between the young cell and each cell, which has been summarized in this figure. One of the way also the epigenetic marks on the histone tail which can be modulated is recently being observed as proteolytic clipping of the N-terminal tail. So as I told you that for histone H3, the 4th and 9th residues are most important, but there have been identification of some proteases where these tails can be clipped off, and once the tail is clipped, so there is the 4th and 9th modification sites are gone, and this clipped histone H3 will take up a different kind of modification to be incorporated or being incorporated, whether it is going to give rise to a heterochromatic domain or a euchromatic domain. Hence, because there are proteolyses which are already been identified in core histones like H3 and H2A mostly, people also searched for histone proteases. So, histone proteolysis started to be discovered in 1976, and as you can see by now, there are not many proteases which are till date identified, and mostly these proteases mostly are identified only for histone H3 and H2A. And even if I listed here many of the proteolysis events till 2021, some of these proteases are highlighted in black and blue, which are only till now identified, which counts to not more than 9. However, if someone looks at the fate of histone proteolysis, they have varied fate, starting from histone proteolysis is observed in foot and mouth disease infection to differentiation, to senescence, to sporulation, conjugation, and many other, mostly differentiation and aging. And even histone proteolysis is also being observed in case of cancer. However, as I told you, that there are not many proteases which are by now identified. So we also had one observation. In case of aged chicken liver, when the total histones were being isolated, it was found that in case of aged chicken liver histones, an extra band appeared which could be probed with histone H3 antibody, and the extra band was not probed with N-terminal anti-acetylated H3 antibody, suggesting that probably it lacks the modification. So when this band was sequenced using N-terminal sequencing, it was identified that indeed it was a clipped product of the histone H3, and it was clipped, and the position of cleavage was identified between 23rd and 24th position from the N-terminus. While this cleavage or while this faster migrating band was seen in chicken liver of aged chicken, the same cleavage was absent in other tissues of the aged chicken such as erythrocyte and brain histones. So, having identified a protease or a proteolysis in chicken liver, it was hypothesized that probably there should be a specific protease which should be responsible for this proteolysis. So when chicken liver nuclear extract was made and it was incubated with total histones from mice liver which do not show proteolysis, in the treated lane the histone H3 disappeared, suggesting that there is a protease in the chicken liver nuclear extract. Subsequently, this chicken liver this enzyme or the protease was purified using an empirically designed purification method, and it was identified as around 55 kilodalton protein or an enzyme which was named as S3H. This protein was sent for sequencing, and through RD-MS sequencing, the protease was identified to be glutamate dehydrogenase, and all the tryptic fragments actually aligned only to glutamate dehydrogenase. No conventional protease were seen in the tryptic fragments. So the question remained, is actually GDS acting as a protease, which is actually a dehydrogenase acting as a protease. So then what was done was we actually then purified chicken liver glutamate dehydrogenase as dehydrogenase using dehydrogenase assay, and after purification we actually tested it for histone protease assay, and we could find that yes, actually the purified dehydrogenase also demonstrated a histone protease activity. At the same time, the peak dehydrogenase activity and the peak protease activity, they coincided to the same fraction, that is in different steps of purification, which was also proven through SDS-PH silver staining and as well as antibody staining, which was raised against the protease, suggesting that at least the peak glutamate dehydrogenase activity and the peak protease activity were actually co-purifying through different steps of purification. Subsequently, allosteric activators and inhibitors of GDH were thrown into the assay system of the S3H assay, and it could be seen that the allosteric inhibitors of glutamate dehydrogenase also inhibited the histone protease assay or the H3H activity, as well as activators, they activated the protease activity, suggesting that at least glutamate dehydrogenase allosteric regulators are also modulating H3H activity in a similar manner. Subsequently, we got an expressing clone of GDH, human GDH as a gift, which was actually expressed, and the bacterially expressed and purified human glutamate dehydrogenase also demonstrated similar H3 specific protease activity, confirming that actually GDH was indeed acting as a protease. Then using the same assay of disappearance of histone H3, the GDH activity and H3H activity were quantified in terms of unit activity, etc., on the basis of the rate of disappearance of the H3 band. And the H3H cleaved H3 products were sequenced N-terminally to find out actually what are the cleavage points at which the H3H cleaves the histone H3, and the points were, and the cleavage points were identified to be, it is responsible for cleaving at the N-terminus and was an endopeptidase which was cleaving between 2nd and 3rd, 4th and 5th, 8th and 9th and 23rd and 24th position of the histone H3. So it was actually the endopeptidase which was acting as a cryptic peptidase having multiple sites. Then we asked a question, if actually the GDH activity and protease activity are existing in the same protein, where are the active sites? So we did very basic biochemical experiments in which we tried to compete the GDH substrates with the H3H activity. The conventional GDH substrates like alpha-ketoglutarate and glutamate could not compete with the H3H activity. However, peptide substrates against the N-terminus of the histone H3 could compete with the H3H activity. And also the ammonium chloride concentration for the H3H activity and the GDH activity were found to be different, suggesting that probably there existed two different active sites for both these activities. And then we asked a question, can we biochemically uncouple these two activities? So whenever the H3H activity was there, it was always associated with the GDH activity. On the contrary, when the H3H was stored for longer in a -70, we could find that the H3H activity was abolished, whereas the GDH activity was fairly the same. So both these proteins of having inactive H3H and active H3H were subjected to CD analysis, and it was found that there was an electricity difference in the inactive H3H, wherein there was a decrease in the total alpha helical content, which was also synergistic, which was also similar to the melting curve assay, wherein the inactive H3H showed that at higher temperature there was protein aggregation, suggesting that there is at least partial denaturation. A map of this sequence and the alpha helical map suggested that the active site for the GDH is not a part of the alpha helix, since our initial data suggested that there is a decrease in the alpha helical content, and the active site for GDH is not a part of the alpha helical content, alpha helix. So it also further suggested that probably the S3H active site was somewhere different from the GDH active site, at least in a different domain. Further analysis also showed that in other tissues where proteolysis of histone S3 was not seen, like brain, heart and muscle, wherein S3H activity was not there, when one of these extracts were added to the active H3H fraction, the active H3H activity of the liver was also inhibited, suggesting that probably this tissue, at least the brain, wherein the inhibition was more prominent, so at least these tissues should contain an inhibitor. So this could be one of the physiological inhibitor. Another group who is also working in this area went further and actually identified the inhibitor, and they identified it to be staphin B, which is actually a cysteine protease inhibitor. And they also came up with a model that why in young versus aged there is more proteolysis of histone S3 mediated, probably mediated by the S3H, and it is due to the sequestering difference of the staphin B in the young versus aged, because in young the staphin
5 more minutes.
Yes, because in young the staphin B is probably expressed in the stoichiometric amount, so they inside the nucleus and the cytoplasm they actually bind to the GDH, and by virtue of that the GDH activity is inhibited. However, in aged cells there is decrease in the expression of staphin B, as well as there is more localization of staphin B in the cytoplasm, so that making GDH free from staphin B and available for H3 to cleave. So that's why probably H3 is cleaved in the aged cell. This also talks about the regulation, how this H3H activity of the GDH is regulated in vivo. Further, they suggested that if even in a young cell, if there is a damage signal, DNA damage signal, it leads to localization of staphin B more to the cytoplasm and GDH inside the nucleus to be localizing more inside the perinuclear area, and by virtue of that there is cleavage of histone S3 in case of DNA damage and control. So then it was hypothesized what will be the physiological significance of the S3 proteolysis. So nuclear fractionation using MNH digestion suggested that probably S3H was located more towards the perinuclear area, which was also confirmed using confocal fluorescence microscopy, wherein the H3H could be staying more to the perinuclear area. And looking at the modification, post-translational modification existing in the cleaved H3, Tomar group had identified that the cleaved H3 contained both signals for activation H3K27AC and inhibition or for inactive chromatin H3K27Me2, suggesting that these are actually probably a part of bivalent chromatin which is having both active mark as well as inactive mark. And secondly, because it was a clipped one, so the clipped H3 also lacked one acetylation site, that is K56, and through our assays we could also find that the delta H3 or the clipped H3 can be proved with H3K36 dimethylated antibody. It is a mark of activation. It is a mark of the active chromatin. So based on these observations, it was proposed that probably the delta H3 is actually making a part of the chromatin of the non-proliferating cells, whereas the intact H3 could be a part of the chromatin of the proliferating cells. In the similar assay systems, actually we have also identified a H2A specific protease, which I am not going to discuss now because of paucity of time, which we are in the process of identifying. And I want to take you to the last slide. Last 1 year or so, we are working on another assay system, I will say, in which what we are trying to do is we are trying to induce aging in mice by injecting D-galactose, and we are rescuing again or inhibiting aging by feeding Tinospora cardifolia. And this is at the preliminary stage. However, I can say that we have already established the assay system, and with this structure, we are able to induce aging in young mice by injecting D-galactose for 60 days, and the aging could be retarded by feeding Tinospora cardifolia. At least we have confirmed it using few oxidative markers which are specific for aging, and we intend to do an epigenetic analysis of at least liver, testis and sperm. What happens to the epigenetic signature, at least in the sperm, so that because it will be actually leading to understand how the epigenetic inheritance will be influenced in the subsequent generation mediated by aging. Thank you very much. So I am thankful to my PhD scholars who have worked in this field, and I am also thankful to my mentor, Professor M.M. Chaturvedi, who is sitting here, and Dr. Anil Panigrahi, who is in right now USA, who actually established the H3H assay system for the first time, suggesting that from the aged chicken tissue nuclear extract, it cleaves histone H3. Thank you very much.
Thank you, Dr. Purohit. It was a very enlightening talk, understanding the dual role of the enzyme as a glucose 3-phosphate dehydrogenase as well as histone H3As. I am sure that there will be many questions from the audience.
Very sensitive on behalf of Dr. Purohit, comrades. I have a concern. Can you hear me?
Yeah.
Yeah. The GDH, and there is we all know that there is a special kind of relationship of the GDH with a special reference to the tissue type liver. There is a purpose in it, right? Because the reversible deamination and the GDH is involved. That is a textbook information. So the question is, I'm wondering whether it is, like the the plant extract, what exactly you were saying it, right? Do you see the modulation of the effect what you are seeing with reference to your aging model? Yep.
Actually, we have not gone up to the chromatin level right now. You are right that your hypothesis is that probably when we are feeding with Tinospora cardifolia, it should also modulate GDH activity. This is what your question is, right?
No. I don't mean that. That is a very straight hit. I don't mean that. I don't think biology would work like that. Because I was wondering, whether it is, because of the special relationship of the GDH for a biological purpose, maybe it could be, I'm wondering, maybe it has got a little to do with the years and more to do with the liver. So if it were to have got to do with the years, because Tinospora, because you have extended lifespan, that phenotype you have seen, if it were to really direct link with the years and this modification of the modulation, so I was wondering at least partially will it be influencing it. That is the logic of what I'm thinking.
Actually, yeah. I take it as a suggestion because, we intend to investigate that. Then only I can,
Any other question? Thank you, Dr. Purohit, for a wonderful talk. So nice of you.
Thank you.